Implementation of a high-dose rate irradiator for living samples using 400 keV electrons. From the MNSU Nuclear Research Team, collaborators include Preston Finger, David Markowitz, Samantha Sundborg, Jackson Walters, and faculty mentor Dr. Andrew Roberts. Direct high-dose irradiation of living samples was successfully demonstrated using extracted high-energy electron beams from the Applied Nuclear Physics Lab Accelerator. The analysis of effects on living samples allows for determination of physical doses delivered. This allows the use of the Applied Nuclear Physics Lab in the future studies of irradiation effects on synthetic and living samples in collaboration with other departments and industry. Electron beam energy and spatial distribution was investigated using magnetic steering and novel detection methods developed with the Geiger-Müller counters. The experiments that followed included the use of radiation badges, fruit flies, and cellular assays to get organic and inorganic methods for measuring dosimetry. The radiation badge experiment suggests a minimum of 120 to 400 grays per hour, a radiation dose consistent with the biological effects demonstrated in the fruit fly irradiations. It was found that this dose rate killed all the fruit flies in 1 hour, while shorter irradiations caused delayed mortality of the population. Work is continuing to develop the methodology of cell assay irradiation to refine dosimetry. A GM counter was placed at varying distances associated with the electron beam travel distance through the air at varying energies. In Figure 7, it was confirmed the accelerator was producing an electron beam. Using the online graphing calculator Desmos, a simulation was created using relativistic electrodynamics to determine GM tube placement to bend the beam from one Geiger-Müller counter to another. Bending of the beam via a magnetic field generated by the Helmholtz coil setup. For measuring dosimetry, the radiation badge was used as a benchmark for the assessment of irradiation for the cell assays and the fruit flies. Experiments were done on all three at approximately the same distances, electron beam energies, and irradiation time for direct comparison. The first fruit fly experiment was conclusive in killing all the fruit fly eggs, while the second had eggs hatch but none matured to adulthood. The cell assay experiments were inconclusive, and further experimentation is ongoing in developing this dosimetry method. These experiments will lead to refined dosimetry rates for this accelerator. With known rates, this lab can be used as a tool in collaboration with other departments and industries. Special thanks to the physics graduate students Henry Lemieux and Daria Hoshenko, as well as our other collaborators in the biology department, Dr. Dan Toma and Dr. Allison Land.